Great. Our final speaker of the morning session is Sangeeta Bhatia. Of course, she's one of our colleagues at the Koch Institute, as well as the director for the Marble Center for Cancer and Nanomedicine. She's going to tell us about activity-based biomarkers for non-invasive cancer detection.
Everyone hear me okay? Yes. Good afternoon. Thank you for sticking with us this morning and for that inspiring talk by Doug. I'd like to really thank the organizers and congratulate them for having this symposium today. Ed and Nancy, I know this has been a real passion project of yours, and it's really exciting to see everyone here today. I'm hoping this morning to share a new technology platform that we invented that we call synthetic biomarkers. And we invented them to try and address some of the limitations that exist in existing molecular approaches to cancer detection. So I'd like to start off by just looking more broadly at the cancer landscape than early detection. And if you think about the sort of life cycle of cancer, there are actually many points along the life cycle, starting at risk assessment all the way through to treatment and recurrence, where one would like better insight into the status of the disease in order to inform clinical management. And in spite of this, we actually don't have very many good biomarkers. So we'd like to assess the risk for cancer. We'd like to detect it early. Once we have diagnosed cancer, we'd like to stratify it for therapy. We'd like to see whether the therapy is working. And then once it's been treated, we'd like to monitor for recurrence. So by and large, the challenges in this space can be summarized by the trade-offs in sensitivity, that is detecting all the cancers when they're there, versus specificity, that is only calling a cancer when there is a cancer there. And you can see, we've heard over the course of the morning, in particular in Dr. Kramer's talk, about the challenges of overdiagnosis. So if you have a very sensitive biomarker, then you can overdiagnose cancer, and then there's challenges associated with that intervention. So why are biomarkers so scarce? For those of you who are reading the literature, you might say to me, well, I see the literature is pouring full of new candidate blood biomarkers for cancer. And in fact, here's just a sampling of some of them. Metabolites, peptides, proteins, cell-free nucleic acids. We heard beautiful work from Dr. Vogelstein's group this morning, exosomes, and even whole cells that are responsible in part for the metastatic spread of cancer. So there's lots of promising candidates. What's wrong with these? So one challenge with these from an engineering perspective is that each and every one of these requires a different specialized analytical tool. And in some cases, these tools exist for work with the blood of cancer patients. In some cases, they need to be invented. So for example, the circulating tumor cell field has really exploded largely because new tools became available to be able to catch and detect those cells. So that's the engineering challenge. What's the biology challenge? Well, the biology challenge from my perspective is that if you're looking in the circulation, you're what Dr. Vogelstein called it biologically limited. You can only ever measure what the body sheds, what the tumor gives you. And that's all you have access to. So you can do correlation studies, and these correlations may or may not be causatively related to the progression of cancer. So in terms of just thinking about how good a blood biomarker can ever be, my group and I have been really inspired by this paper that Sam Gambier published about 5 years ago now. And I have to say, as I can understand why many of you might not be inspired by a computer simulation, but that's what this paper was. But engineers are inspired by computer simulations, and I want to tell you why. This was an amazing computer simulation. So what Sam and his group did was ask the question, how good could a protein blood biomarker ever be? And they did this in the context of ovarian cancer. Ovarian cancer, as we know, has a vast discrepancy for survival rates when it's detected early versus late, and over 75% of patients today are detected late. So you can take a blood biomarker like CA-125 for ovarian cancer and ask, how good could it ever be using analytical tools that exist today? And the computer simulation was, imagine how fast a cell can make a protein, how fast it can diffuse into the blood, what the background from normal tissues would be, and how long that protein would stay around in the circulation. And let's compare that to what we know about typical tumor progression rates and ask how soon could we ever detect a cancer using a protein blood biomarker after tumor initiation? And the answer is very sobering. It's 8 to 10 years. Okay, so it's quite a long time. And in Sam's paper, they outline really what the challenges are to making that time shorter. So first of all, the background of the secreted biomarker by healthy cells is limiting. Second of all, blood is actually, from an engineering perspective, a horrible matrix in which to measure things. It's full of billions of cells. It has all kinds of proteins that would foul your sensors. And so it creates a noisy environment. And finally, the blood pool is 5 liters. So whatever is being shed by the tumor is being diluted into 5 liters of blood. Okay, so taking all these kind of into account to sort of summarize what the current biomarker paradigm is, we think they're endogenous biomarkers that are biologically limited. Typically, they have been historically single parameter measurements, which lead to the difficulty I mentioned earlier between sensitivity and specificity. And finally, they're in blood. So we'd like to sort of disrupt this with this new idea that we're calling synthetic biomarkers. You can sort of bear with me because if you haven't heard this before, it might sound a little crazy. And the basic idea is to deliver a probe, okay? Deliver a probe, a nanosensor, that will make a measurement that's relevant to the cancer biology. Okay, so it's an exogenous probe. Now, if you design this probe and you deliver it to the patient, it's actually not so unusual. There's a precedent for this clinically where we give radioactive glucose analogs to do PET imaging scans and look at where glucose uptake is in the patient. So here we're doing something similar. We're giving a probe, we're making a measurement, and I'm going to show you that we're going to make the measurement in a body fluid instead of doing an imaging scan. Okay, we design these probes so that we can overcome some of the sensitivity limitations that I mentioned earlier. In particular, we make them enzymatically activated. And enzymes, as you know, don't get consumed in chemical reactions. So they can catalyze a reaction over and over and over again. So you have a naturally amplified process. We're going to make multiple measurements. So instead of just one parameter, we're going to multiplex the assay. And finally, we're going to design these probes so that when they find their target, they deliver a signal in a better matrix than blood, and that's urine. So urine is the body's natural filtrate. It's concentrated about 20-fold from the blood. In a normal patient, it will have very low protein background and very few cells. So it's a better place to measure things. So that's the basic idea. What enzymes will we measure? Well, we're very interested in measuring this family of enzymes, endoproteases. So there's about 566 of these proteases in the genome. They measure endo within, they cut peptide chains within a peptide chain. They're roughly half extracellular and intracellular, and they're classified by their active site. And what's interesting about this class of enzymes to me, having trained as a physician, is that all of the architectural change that we learn about in histopathology, whether it's carcinoma in situ, whether it's invaded, whether it's metastasized, all of the architectural change that a histopathologist sees in the microscope is really driven by this class of enzymes. They are responsible for remodeling the extracellular matrix that leads to cancer invasion and spreading. So we think they're centrally linked to cancer progression, and in fact, if you look in the literature, you can find a whole list of endoproteases that do seem to be involved with different stages of cancer progression. I'll highlight this particular one, matrix metalloproteinase 9, because I'm going to refer to it later in my talk. Okay, so that's the basic idea. We're going to create synthetic biomarkers. We're going to try and make them sensitive, specific, and hopefully, based on the last few talks this morning, medically actionable, so that they can inform things that we could do in the clinic. I'm going to start with a cartoon. I apologize, it's highly stylized. It was made for a different venue, but I think it's useful to get this sort of idea across. So first, let me talk to you about sensitivity. So this is a cartoon of a solid tumor around a blood vessel, and we're going to be injecting the nanosensors in the circulation. These nanosensors are about 50 nanometers in diameter, and here you'll see them extravasating through the hyperpermeable vasculature of a tumor that's undergone the angiogenic switch, which happens around 1 to 2 millimeters in size. So these sensors have been designed them to enter into the tumor interstitium and be sensitive to endoproteases that are released as part of the tumor biology. Here you're going to see an artist's rendition of endoprotease cutting, catalyzing 1,000 chemical reactions an hour. So you can see there's an amplification process in the sensor. Molecularly, this is actually 1,000 cleavage events. Now we've released these fragments from these sensors in the tumor interstitium. And so the question is, how do we get this activated sensor out of the body to act on it? And so here what we do is we use the fact the kidney, as I mentioned, is a natural filter. The kidney is a 5 nanometer filter. So if we produce cleavage fragments that are smaller than 5 nanometers, the parent particle will not be filtered into the urine, but the smaller fragment, the cleavage fragment, will be filtered into the urine. Okay, so if we put this whole thing together, you'll see the parent particle is delivered intravenously. It enters into the tumor interstitium where it can be activated by endoproteases ultrasensitively and amplifying that reaction 1,000 times per enzyme. And the fragments then are liberated, filtered by the kidney and into the urine. Okay, so as an engineer now, we have an amplified signal against a measurement we wanted to make about the tumor biology in the urine. Okay, so that's the cartoon. What about data? So this idea has sort of two key pieces. It has to have enzyme-activated amplification cutting reaction, and it has to have this pharmacokinetic switch, which is that the parent particle doesn't get out, but the fragment gets out. So here are some data on these. So this is an enzyme sensor. This is our first generation sensor. So in the beginning, we made these with an iron oxide core, which you see here. So this is a standard MRI contrast agent. And conjugated on the surface of that core is a chain of amino acids, which you see here, that have been pulled from the literature to have a cleavage motif for the protease of interest. So this is a thrombin-sensitive enzyme sensor. And in this experiment, what we've done is we've made a fluorescent-on version of this sensor. So when you add thrombin to this sensor, you can see that the fluorescence turns on. And importantly, when you add a variety of different endoproteases that do not recognize this motif, the sensor does not turn on. So we've made a relatively specific sensor against the enzyme of interest. And you can see that this is an amplification process. So it's a sensitive sensor and a cleavage fragment. So now the question is, does the parent particle stay in and the fragment get out? This is a mouse experiment where we have a fluorescent version of the sensor that's liver targeted. So you can see in the control animal, the particles accumulate there. And in the diseased animal that elaborates an endoprotease to which this sensor has been designed, the particles target the liver, they get activated, and this cleavage fragment gets liberated, filtered by the kidney, and accumulates in this target organ here. That's the bladder. Right, so that's the urine accumulating in this animal. So we have a sensitive sensor and a pharmacokinetic switch. And we then wanted to think about sort of copying Sam's experiment, both theoretically and experimentally, and ask the question, could these be used for early cancer detection? So in order to look at cancer detection, we nominated several proteases from the literature. And this is just an example of one that I'll walk you through. So this is MMP9, matrix metalloproteinase 9, which is a type 4 collagenase. So type 4 collagen, of course, is in the basement membrane of epithelia. And you can see if you just look at the cancer genome atlas at the message level that this is overexpressed in a variety of tumors. If you look on a human tissue microarray of colon cancer, you can see that it's also found at the protein level. And in collaboration with Richard Hines at the Koch Institute, who's developed a new method for proteomics in the extracellular matrix of tumors, we also find MMP9 in human tumor colorectal samples that's bound to the extracellular matrix. So it's present there in the interstitium of tumors. So we designed a nanosensor against MMP9 and set out to test it in human xenograft models. Now, we wanted to compare ourselves against a blood biomarker, as I said, to be inspired by this study. And the existing blood biomarker, which is FDA approved for colon cancer, is actually not very sensitive or specific, and that's CEA. But nonetheless, it's what we had to work with. So this is an old blood biomarker that was first described in the 1960s, and it actually only picks up about 25% of early cancers. And if you look at human colorectal cancer cell lines in vitro and look at levels of CEA production, you can actually see that it varies by a couple of log. So it's not surprising then that patients would have highly variable levels of CEA. And in fact, this is one of the limitations of some of the shed biomarkers that are not intricately linked to the cancer biology, is sort of you get what nature gives you. Okay, so in order to do our experiment, we wanted to make human cancer xenografts in mice using the best case scenario. So imagine that you had a tumor that's making a lot of CEA, and you make an MMP9 sensor, and you ask, how do you do against that blood biomarker using the synthetic biomarker system? So here's the experiment. So here's the mouse with the tumor, and you can see again, just as the quick check, that the bladder is lighting up when the animal is tumor bearing as compared to the controls. We can see that the urine fluorescence comes on here on day 10, and this can be inhibited with a broad spectrum MMP inhibitor to show that this is specific for MMP9 activity in the cancer, in the tumors. And then finally, you can sort of quantify how good your biomarker is. Some of you will be well familiar with these plots, but I'll just describe them for a second. This is what we call a receiver operating characteristic curve. And a random diagnostic would be along this diagonal line, so that would be an area under the curve of 0.5, and a perfect diagnostic would be an area under the curve of 1. Typical promising diagnostic, then the preclinical development would be 0.8, something that's not so good but still in people like prostate PSA is like 0.67. So here you can see CEA on day 10 of this experiment. So the blood levels of CEA in this animal are 0.61, but the urine test is 0.94. So that's promising because we can do better than a best case scenario. But of course, the most exciting thing would be able to detect tumors in patients that don't have CEA levels. So here now we grew a tumor that had low secretion levels of CEA. And you can see when we look in the blood of this animal that the CEA levels never come up. But yet, on day 13, we're still able to quantify a robust urine fluorescence when we give an MMP9 nanosensor probe. These tumors are about 5 millimeters in size at the time that we're able to detect them, and we're quite excited about that result. So then to sort of ask the question theoretically, which is how could this be in people, to sort of mimic Sam's work, we went on to do our own model, which I won't walk you through because I know none of you will appreciate it. It's a beautiful study done by Gabe Kwong. We developed this model. We reformulated these particles from an iron oxide core to a polyethylene glycol, so an inert polymer that we felt could be translated to patients. And then we asked in patients that were high CEA producers, if they had a 1 centimeter mass, how good could your blood biomarker be relative to the urine detector? And here you can just look at concentrations, just analytically, the CEA levels here are in sub-nanomolar, and we get about an 80-fold amplification in the urine in terms of concentration of analyte just from using this approach in humans. So this is quite exciting. The model, incidentally, also tells us how we can improve the technology, and Esther Kwan and Jaydeep Dudani in the lab have gone on to develop a targeted version of this nanoparticle, which works about 10 times better. And in unpublished data, we're now detecting ovarian cancer lesions that are as small as 2 millimeters. So that's the story on sensitivity. We think that we can get sensitivity using this approach by signal amplification. And I'd next like to turn to this idea of specificity, which is not having false positives. So how might we do that? So I already sort of told you that we're going to do this by multiplexing, which is that we want to make multiple measurements. And so our idea for how to multiplex this technology is to actually give not just one nanosensor probe, but to give multiple nanosensor probes. Okay, and the idea is that each one of these has a different peptide motif that makes it sensitive to a different cutting event, a different protease, and that the liberated fragments will have a unique mass. So they're mass barcoded. So you give a cocktail of these. We typically give a cocktail of 4 to 10. They go to the site of the tumor, they're activated, the liberated fragments get concentrated in the urine, and then you mass spec the urine to identify each of those fragments. That's the basic idea. Okay, and so you might think this is for the mass spec aficionados. I won't walk through the specific strategies, but this is sort of a typical 10-plex barcode being mass spec for us. So we take this mass spec library, and then the question is, what 10 endoproteases do you measure, and where might this improve the technology? So here, we've been very interested in the idea of lung cancer detection. It was mentioned earlier that there's a new recommendation for lung cancer screening in high-risk patients. people who have a long lifetime history of smoking. And the challenge with this, the screening recommendation has been that about 24% of screened patients have positive results. However, 95% of them are false positives. And the reason for this, if you sort of look at the lung parenchyma radiographically in lung cancer as compared to chronic obstructive pulmonary disease, COPD, or an interstitial lung disease, in fact, many of these may look initially similar on a screening study. So the question is whether the endoproteases that are elaborated in these diseases may be different, and we may have a tool now to stratify cancer versus not in these patients. So to ask this question, Andrew Warren in the lab did a genomic analysis, first looking at the Cancer Genome Atlas for lung adenocarcinoma, and he came up with a list of endoproteases that are robustly overexpressed in lung cancer. And then he also looked in the Lung Genomics Research Consortium database against COPD and interstitial lung disease. What was exciting about that was, in fact, that he could find classifiers. This isn't projecting that well, but basically these yellow lines, MMP12 and kallikrein-6, were robust classifiers that stratified tumor and could call out interstitial lung disease and COPD. So by virtue of combining these multiple biomarkers, you're now able to make a sensitive call, and we think a relatively specific call. So to ask whether this would work and how sensitive it would be, we collaborated with Tyler Jacks, who's my suite mate. I have a great suite. To use one of his fantastic mouse models. So this is a mouse model of non-small cell lung cancer, which works by inhaled adenoviral Cre recombinase, and it creates a p53 knockout and a mutant KRAS lung cancer. So we start those experiments at time 0. We then go on at 2 weeks to start doing this injection and then urine testing. If we look in the lungs of these animals, we can see now we're actually delivering the synthetic biomarkers by inhalation, and they get right down into the lung parenchyma and all the way up to, all the way up to the tumor edge. And as the tumor progresses, you can start to see the urine fluorescence come on around week 3 and robustly around week 5. And if you now ask if this is a good classifier, by week 5, we have a perfect prediction for tumor-bearing animals. And again, we can see by CT, these are a few millimeters in size at this stage. So we think this is quite exciting for the potential to stratify radiographic findings in lung cancer. But another opportunity that came up earlier today, which we're excited about, and we recently partnered with Janssen to start working on, is actually once you have a tumor, so imagine you have prostate cancer, stratifying an aggressive, worrisome lesion versus an indolent or slow-growing lesion. And the hypothesis is that these will, in fact, have different protease patterns because the tumors are differentially aggressive. And that's something that we're, sort of an application that we're now working on. So I've told you about sensitivity and specificity by multiplexing. In the last few minutes, I'd like to turn to how one might make these findings actionable. So the first example I'd like to give is an example of liver cancer. Some of you in the crowd know that half of my lab, I spent most of my career trying to build an artificial liver. The livers that we're trying to build are to replace failing livers, primarily due to hepatocellular carcinoma, which is driven by several etiologies. This is actually a poster child for for cancer prevention. It turns out that hepatitis B vaccine was the original anti-cancer vaccine. It prevents chronic hepatitis B and hepatocellular carcinoma. And the recent hepatitis C drugs, the potent antivirals that you've heard about, if they're given before late-stage cirrhosis, they also largely prevent hepatocellular carcinoma. So these are interventions that can prevent liver cancer. However, there's a new offender, and this is a disease primarily of the developed world, which is driven by obesity, and that is fatty liver disease. And fatty liver disease leads to non-alcoholic steatohepatitis, which can lead to cirrhosis and then cancer. And 40 million Americans are at risk for this disease. The current therapies are only weight loss and bariatric surgery. But there are 50 drugs in the pharma pipeline, the first of which are thought to be hitting in the next couple of years. So soon there will be a drug for this form of cancer risk. And the question that we've been very interested in is how do you decide who is at risk for this and who would need drug? So we thought maybe we could stratify patients that were at risk for liver fibrosis using our technology. And it turns out that fibrosis progression is also driven by endoproteases. So we collaborate with Detlef Schupan at the BI Deaconess Medical Center on this model, this chemically induced model of liver fibrosis, made a 10-plex and shown in this animal model that we can perfectly predict the animals that had liver fibrosis in this setting. Okay, so that's one example of how you might prevent cancer by using a tool like this. Another example is that there's a set of therapeutics that are emerging. These are protease-activated antibodies and other drugs. There's a set of these, for example, in the immuno-oncology space, and the question is which patients have a high proteolytic background that should be given these drugs. So we think we might be able to stratify patients like this using this technology. And finally, the question comes up, so okay, great, once the urine test is positive, where is the tumor? And so we've been working on a version of this that could be administered subsequently, which would be active on PET. So this is a PET active version that's been developed by Liang Hao in the laboratory, and in some settings this could be a stage for surgery. So those are some medical actions that we're hoping could come about. In the last couple of minutes, I'd just like to touch on a sobering result, the sobering sort of observation, which is that 70% of cancer deaths by 2020 are actually going to be in the developing world. And in thinking about that, we had developed a mass spec-based tool, which is not going to be available here. This is an image from a student of mine that just traveled to Botswana for a medical rotation. This is the National Diagnostic Lab in Botswana. And so we started thinking about how might we make our tool available in that setting. And we were inspired by what's been done for HIV detection in that region, which is paper diagnostics. So because we have a synthetic probe and we can design the fragment that comes off to be barcoded in any way we want, instead of just getting what the biology gives us, we design them now to be trapped on paper. So now we have a paper diagnostic version of this multiplex barcoded synthetic biomarker. And we imagine that one could even take a picture of this on a smartphone and sort of send it to a remote provider in the future. So just to sort of summarize, I've told you about the synthetic biomarker platform that we invented and we're excited about. I've told you about how it can be sensitive by signal amplification, how it can be specific by multiplexing, and some ways in which we think it will be medically actionable. I've shown you proof points in colon cancer, lung cancer, and liver fibrosis in mice, and we have other examples that are emerging in the lab that we're quite excited about. And finally, we just started a little baby startup to try and take some of these ideas towards patients. So I'd like to thank you for your attention. This is the group who does all the work. It's lovely to be at the KI where this work can be accelerated, and our generous sponsors. Thank you.